Assalamu alaikum everyone. Welcome to another ATP video. In this video we'll talk about some basics in microbiology to get you introduced to this subject. So, what is microbiology? Microbiology is the study of microscopic organisms, such as bacteria, viruses, fungi, and protozoa. Most of these microorganisms are beneficial to us. In humans, we have normal microbiota that helps in digestion. In plants, the bacteria is responsible for nitrogen fixation. And in animals, it helps in cellulose digestion. Also, these microorganisms are used in industries. Generally, we divide the organisms, these bacteria, viruses, and fungi, according to their characteristics. So in bacteria, we use gram stain, either positive or negative, to categorize them. In viruses, we depend on their genome, DNA or RNA, whether it's single-stranded or double-stranded, to categorize them. In fungi, they're either divided into molds, which is multicellular, or yeasts, which are unicellular. We also have the hierarchy of classification. We used to put each organism, including us humans, into its category. We took it in high school, if you remember. So from highest to lowest, it's domain, kingdom, phylum, class, order, family, genus, and species. You can use this mnemonic to help you remember it. Dina's kids prefer candy over fried green spinach. Spinach is pretty underrated, by the way. So if we start from the top, we have three domains on this planet: bacteria, archaea, and eukarya. Both bacteria and archaea are considered as prokaryotes, whereas eukarya are considered as eukaryotes. The main differences between prokaryotes and eukaryotes are absence of cytoplasmic structures such as mitochondria, Golgi apparatus, and endoplasmic reticulum in prokaryotes. And then we have absence of cell wall in eukaryotes except for fungi and plants. Also, they differ in the structure of the ribosomes. After that, we go to the kingdom. Both bacteria and archaea have only one single kingdom, which are bacteria and archaea, respectively. And then we have the eukarya, and it has four kingdoms: protists, fungi, plants, and animals. And then we continue the classification like this, like we mentioned before: phylum, class, order, family, genus, and then species. For now, we will focus on bacteria. How do we name them, basic structures, and gram staining. Any bacteria's name is composed of two words. The first one refers to the name of the genus, and the second one refers to the species name. We'll take Staphylococcus aureus as an example. So Staphylococcus is the genus, and aureus is the species. Also, the name of the microorganism might refer to its shape, like in our example. Staphylo means a bunch of grapes, or a cluster, and coccus means round cells. Aureus means it looks like gold in color when we culture it. Other than cocci, the round cells, we have rods, or bacilli. And then we have spirochetes, which are curved. And then we have diplococci, which is basically two cocci, or basically two round cells. And then we have coccobacilli, which is a mixture of both a rod and a round cell. And then we have vibrio, which is a curved rod. Then we have streptococci, which are cocci in chains. And then we have the staphylococci, like we mentioned in our example, which are large cocci in irregular clusters. And then we have tetrads, which are cocci in a packet of four, from the name tetrads, tetra, which means four. Now we're going to discuss the bacterial architecture. Since bacteria are considered as cells, that means they have genetic material, a cell membrane, and a cytoplasm. In addition to that, they have a cell wall, a flagella, which is used for locomotion, and then we have pili, which is used for attachment, and then we have plasmid, which is an extra chromosomal genetic material, so it's not part of the DNA. And then we have a glycocalyx, which is used for protection. The glycocalyx can be either capsule, which is neatly organized and protects the bacterium from phagocytes, or biofilm, which is an unorganized, loose structure, yet important for attachment. Bacteria also have porins. These porins are transmembrane proteins that selectively allow some molecules to pass the cytosol of the bacteria. Porins can mediate antibiotic resistance by inhibiting some antibiotics from entering the bacteria. They also have mesosomes that are equivalent to mitochondria in eukaryotes. Remember, bacteria doesn't have mitochondria, so they have mesosomes instead. Bacteria depend on these mesosomes for respiration. Mesosomes are formed by the invaginations of the plasma membrane into the cytosol. Generally speaking, this is how a bacterium looks like. However, each bacterium has its special characteristics that distinguish it from the rest. Now we'll move on to gram staining. In gram staining, we can classify bacteria into either gram-positive or gram-negative, and that depends on the cell wall constituents and its ability to react with the gram stain. To classify them, gram-positive react with the stain and give blue color, while gram-negative react with the stain and give red or pink color. If we look at the membranes of both gram-positive and gram-negative bacteria from the inner layer, we see that both of them have plasma membrane. However, on the outside, the differences start to appear. In gram-positive, it has a thick peptidoglycan layer. This layer has teichoic and lipoteichoic acids. In gram-negative bacteria, and outer to the plasma membrane, we have thin layer of peptidoglycan, and then we have outer membrane composed of phospholipids that has transport proteins and lipoproteins embedded into it. And the last layer in gram-negative bacteria is the lipopolysaccharide. The lipopolysaccharide layer is composed of three structures: O antigen, core polysaccharide, lipid A, which is also known as endotoxin. So we can conclude that the endotoxins are only present in gram-negative bacteria. These endotoxins are antigenic, which means they can elicit an immune response, causing some symptoms of fever, weakness, aches, or shock to the patient. So, that's it for the intro. Hope you enjoyed. Please give us your feedback so we can improve in the next videos. And don't forget to like and subscribe to get our latest new videos and explanations.
Armando Hasuringan, Biology and Medicine videos. Please make sure to subscribe, join the forum and group for the latest videos. Please visit Facebook, Armando Hasuringan. In this video, we're going to talk about the mode of action of antibiotics. So we're not going to look into extreme detail, but we'll just look at the overview in a way. So mode of action of antibiotics. Now we have to know that antibiotics either destroy or slow down the growth of bacteria. So therefore, if it destroys a bacteria, it is bactericidal. If it slows down the growth of bacteria, it's bacteriostatic. Here I am drawing a typical bacteria with cell membrane and cell wall. The cell wall and cell membrane structure I am drawing is characteristic of a gram-positive bacteria, but we will just assume it is any type of bacteria. Within the cytoplasm, within the bacteria, we have a circular DNA. We can have RNA being synthesized and ribosomes here. Ribosomes will synthesize proteins. The first type of antibiotic are the ones that I want to talk about are the ones that target the cell membrane. These antibiotics will disrupt the cell membrane function, which means that they disrupt the phospholipid bilayer. So here is the cell membrane and here is the cell wall, known as a peptidoglycan. They are different. The antibiotic binds here to the cell membrane. It will alter the cell membrane structure and will make it more permeable. This will just disrupt the osmotic balance, causing leakage of cellular molecules, and essentially it will increase the water uptake, leading to cell death. Examples of these drugs are polymyxins, which I spelled wrong, and polines. Now, polines are actually specifically used against fungal pathogens, but that's just a note to know that you can use polines that target cell membranes in fungus. So polymyxins and polines target cell membranes. Other antibiotic classes target the cell wall. These antibiotics will inhibit cell wall synthesis, so these drugs are therefore bactericidal. They will cause bacteria to die. Example of these drugs are penicillin, cephalosporin, and bacitracin. I hope I pronounced that right. All these drugs have different mechanisms of action when it comes to inhibiting the cell wall to synthesis. A good thing about these drugs is that it won't affect human cells because human cells do not have peptidoglycans. We do not have cell walls. Penicillin and cephalosporin are beta-lactams. And then you have the bacitracin and other glycopeptides that prevent the synthesis of cell walls as well. Antibiotics that disrupt the cell wall is pretty extensive, and so hopefully I will make a video on that. Cell walls are important to bacteria. Disrupting the cell wall or preventing in cell wall synthesis will result in cell death. The next type of antibiotic class is the ones that can inhibit here DNA synthesis, or what's occurring here, RNA synthesis. So these antibiotics will inhibit RNA and DNA synthesis. So here the bacteria are typically having DNA replicated, of course, like so. So we have antibiotics that prevent DNA from being synthesized. These are quinolines and also a class known as nalidixic acids. And then, of course, RNA can be made from the DNA. And there are drugs such as rifamycin, which prevents RNA synthesis and therefore protein synthesis. Other classes of antibiotics also inhibit protein synthesis itself. Protein synthesis is carried out typically by ribosomes, which translate mRNA into proteins. Bacteria need to make proteins in order to survive. However, there are antibiotics that target either the 30S subunit or the 50S subunit of the ribosomes. Erythromycin and chloramphenicol target the 50S subunit of ribosomes, whereas there are tetracycline, streptomycin, and gentamicin that target the 30S subunit. Either way, this will disrupt the ribosomes from making proteins, which will result in the bacteria obviously unable to make proteins, so the bacteria is pretty much useless. Thus, the bacteria can still be alive, but it just is unable to make anything. And so, these antibiotics that target protein synthesis are bacteriostatic. They prevent bacterial growth. The last type of antibiotic are the ones that inhibit folic acid metabolism. So normally, this green thing here, called PABA, which stands for para-aminobenzoic acid, it's a precursor to folate. So the bacteria can metabolize PABA into folate. There are antibiotics out there that inhibit folate or folic acid metabolism. See, PABA normally can be metabolized to folic acid within the bacteria, and folic acid or folate is essential for the synthesis of adenine and thymine, two of the 4 nucleic acids that make up DNA. These antibiotics such as sulfonamides and trimethoprim prevent conversion of PABA to folate and therefore prevent proper DNA synthesis, you can say. Humans do not synthesize folic acid, and so these antibiotics are okay to take for humans and are selective towards bacteria. I hope you enjoyed this short video on antibiotics. Thank you for watching.